Hello, everybody. Good morning. Thank you for joining us today. We are excited to have you here for this exclusive webinar featuring Athos Therapeutics and Celaris Bio, where we will share a real-world end-to-end example of how Microtech cell target engagement platform is applied to accelerate drug discovery. We have two excellent speakers. We have Dr. Iordanis Karagiannidis and we have Dr. Ivan Babik. A short introduction here. Dr. Iordanis Karagiannidis is the Chief Scientific Officer at Athos Therapeutics and has been with the company since its inception. He brings deep expertise across academia and industry with focus on targeted small molecule therapies in cancer and inflammatory diseases, including IBD. Following his training, PhD training at Boston University School of Medicine and postdoctoral training at Harvard Medical School and faculty leadership at UCLA. At Athos, he oversees all in vitro and in vivo preclinical research, integrating data to drive IND-enabling strategies and advancement towards the clinic. Dr. Ivan Babik of Celaris Bio is co-founder at Celaris Bio. Prior to Celaris Bio, he was assistant professor at John Wayne Cancer Institute and has authored over 30 scientific publications. He has over 20 years of experience in preclinical and clinical drug development and is inventor of Microtech cell target engagement technology for high throughput drug screening and discovery. So we'll get started. At the end of this webinar, we will have time for questions and answers. So please feel free to submit your questions through the chat. So let's get started. Dr. Karagiannidis, it's all yours.
Thank you, Elmar. And despite what my background says, I'm still the CSO of Athos, and hopefully will be for a long time. And I want to make clear in the beginning that this is not to present what Athos is a company. This is to present how Athos was in the need of help from Celaris in order to promote one of our programs to a later stages of preclinical development. So all the early information on Athos will actually create the narrative of why Celaris is and their methods and techniques are very helpful in companies like ours that are in drug development. So Athos is a clinical stage biotech company, pioneering the development of artificial intelligence-based precision therapeutics for autoimmune disease and cancer. And so we are a dual threat company. Basically, that's how we like to think of ourselves as we have typical drug discovery area, and we're also having a software development platform that are under the umbrella of the same company. So how do we go about discovering our targets for therapeutics? We like to present here how we integrate several big data information in order to create the information that will provide us with the appropriate direction actually to lead to these discoveries. With this approach, what we do is like we bring together big data analysis of exposome, genome, microbiome, immunome, and other omes. In the beginning, early on now, at this stage of our development, we do not integrate that many omes together. We're at the stage where we can integrate 2 or 3 omes. But what we hope to do is bring all this information together and lead to primary mechanism, molecular classifications that will identify targets, and later on in the development to even personalized therapy through analysis of the demographics of patients. I cannot find my, okay. So the order of things for our drug development platform is that we identify the targets in an AI, machine learning, unbiased uh, method which targets the entire disease interactome. And when we're talking about interactome, we described in the previous slide that we are using several omes that are portraying what is happening within the cell or the organ or the organism at the same time at different levels. And then after we identify our targets, and we'll do a brief introduction of how we identify them, we move on to small molecule development. We have an internal chemistry team that develop these compounds where we do some routine studies that help us identify the lead compounds. Then we move on with our preclinical evaluation on multiple models of disease. And usually we focus on autoimmune diseases in our company, but we're not limited there. I mean, depending on the targets we come up with, we are also flexible to, for example, we have a program that is also in leukemias. Then hopefully we move on to clinical development where our targets of choice and the molecules of choice move on to the GLP studies and hopefully phase 1 clinical trials. And our lead program right now, actually, we are in the process of applying for an IND for phase 2. And this is how we get to our targets. We combine all the OMs and we are basically getting a bird's eye view of the whole interactome. And you can see how mistakes are done in the traditional approaches, where people were focusing on hot molecules or popular molecules that may have been finding themselves in the edges of the interactome. What we're looking for are the molecules in the middle, like the AD drug in blue here. We want to target that central hub. I don't know if you can see my cursor, but on the graph to the left here at the bottom, you see the blue X sign. And this is the type of molecule we want to target. The molecules are in the middle of the interactomes and they can affect most of the processes. So there is no compensation through mechanisms that are in the periphery. This is the example of our lead molecule. And again, I'm going only through the surface of what we're doing here to give you an idea of how we approach this, since this is not the focus of this presentation. The focus here is that we did find our target. We integrated the OMs, epigenome, transcriptome, and cytokinome. In this case, it was only the transcriptome, cytokinome, and we got our analysis, and our analysis showed us the top networks involved in this disease. In this particular case is IBD patient biosamples, which we took in-house in our biobank or bought the information from institutions that do this out there, like the CCF. After we analyzed all the data together, we came up with the top networks that are differentiated in this disease. And this is where our personal input comes in. So we chose the networks that had high p-values and high net scores. However, we found the molecules that are in the middle of these networks. We chose the molecules, the enzymes. And in this case, we chose I don't know if you can see it here, but it's EHMT2 or G9A. It's a methyltransferase that is in the middle of this whole hub and of IBD patients. And we found actually that this network is more prevalent in subtype A patients, which are active UC and CD patients with moderate to severe disease state, colonic disease location, and most importantly, anti-TNF and anti-integrin refractory disease. So we moved on. This was a very interesting target. We moved on with it. And we saw that there are two sub-networks within the big network that are very important to us. And this is why we picked this target. One network has to do with Treg differentiation, which is a very strong anti-inflammatory network that promotes Treg immunosuppressive function. And the other sub-network involves tight junction regulation of the epithelial wall, which is very important in this disease. So this is a central hub molecule that is a dual threat. It affects two very important processes of the same diseases. And this is our compound, and this is our actually the labs. This is where our drug discovery labs are located. We also have another location that helps develop our platform. So all of our thought process, all of our approach here will be translated through our platform. And this is the whole approach again. After we discovered our targets of interest, we moved on with computational chemistry and created a list of compounds that could hit the target. Then our medicinal chemistry group created these compounds. Our analytical chemistry group tested the physiochemical characterization of these compounds. We did cellular studies to prove the efficacy at the in vitro model. And then we did a lot of animal studies to prove the efficacy in the vivo models. And this is what led to our lead compound, which is Athos 63, and which is now, which successfully finished phase 1 studies and is now close to initiating phase 2 studies for ulcerative colitis. Oh, this is okay. So 91. Why 91? Oh, I'm missing a slide here, I guess. 91, and I will give you the information here. Why Solaris? 63 was a great compound. It actually fulfilled all our characteristics that we needed to take it to the clinic. However, people familiar with these programs know that such compounds require backups. And 91 was created within the same program. And because it has a lot of the similar characteristics, it performed great in all of the analysis that we did, including the vivo efficacy studies. We hoped it was hitting the same target. And since we outsource the method of target binding, the results from the target binding came later, and Athos 91 was not hitting our target, the desired target. So we had a great molecule in our hands, and you'll see from these studies, it had very good tolerability in mice. It did not, in the HERC studies, it was very safe. We did both colitis efficacy models, and it showed great results. And as you can see here, both in the DSS and TMBS models of colitis, among other studies, we did solubility, stability, all of the metabolic studies. There were no incriminating metabolites. This was an excellent compound, excellent compounds that did everything 63 did. It was a ready program to go to GLP, but we had no target. And this is a very unconventional way of coming up with great molecules, but it happened to us. And as you can see here, look at the pathways. It's hitting all the immune pathways, IL-17 signaling, rheumatoid arthritis, TNF signaling, all the great JAK-STAT signaling pathways. Those are all involved in IBD. So we were so happy we had our backup, but it was not a backup. So we started looking. We looked at 15, more than 50 known receptors and ion channel proteins without any success. We looked at over 400 kinases without any success again. We looked at the remaining methyltransferases. And you know the result again, the acetylase panels, everything we did And among others, we did single assays also. And upon repeated failures, we outsourced target discovery to two CROs, one in Canada and India, for traditional target convolution methods. I'm not going to mention the names here because it's irrelevant. We wanted an unbiased target identification of Athos 91. So we wanted to pull down everything that comes out down with Athos 91 and then identify it. This was an abject failure. So knowing Elmar from previous professional interactions, he contacted me when they started Solaris and he said, we have this method here to help you identify targets. And we gave them a chance, and both Ivan and Elmar here will explain to you how we found our target, and we actually are extremely glad with it. And we now have a new GLP-ready program just because we had the help from Solaris to identify this one, the important detail. And also, I would like to point your attention that you see what we're doing, creating a platform that uses multi-omics in order to integrate big data to bring information, like massive information to us in a short area where we can analyze it. I think Solaris, with their program, this can be integrated to such platforms as a complementary program. So you should keep that in mind as a complementary program to accelerate high throughput type of approaches like ours. And I pass it to you now.
Thank you, Jordan. That was a nice overview of some of the things that Athos Therapeutics is doing. What I want to focus the webinar right now and what I'll talk about right now is Solaris Bio's microtag cell target engagement platform and how the platform was used to help Athos Therapeutics identify and validate some of the potential protein targets of 91. So the microtag system, it's based on enzyme complementation and is composed of two subunits. One is a small 15 amino acid subunit that we call the microtag, and that tag can be cloned at the C or N terminus of any protein target. And the second subunit is a large subunit we call the microtag binding protein. So when the microtag binding protein associates with the 15 amino acid microtag, they form an active RNase enzyme. And as a substrate, we add a small stretch of RNA depicted in panel B that is flanked by a donor and a quencher fluorophore. So when that RNA is intact, the donor and quencher fluorophores are in close proximity and there's no fluorescence. But when it's cleaved by this active RNase, then you get separation of the two fluorophores, resulting in fluorescent signals. So that's basically the system, how it works. Now, we use this system with thermal shift assay as a means to assess the binding of a drug to a protein target within cells. And so with the thermal shift assay, combining it with the microtag system, what's involved is overexpressing a protein target that we've tagged with the microtag in a disease-relevant cell system, applying heat in the presence of potential ligands. And what the heating does is it causes that protein target to unwind, denature, and form insoluble aggregates. And as a result, that 15 amino acid tag that we've clonally added to the target becomes buried in those insoluble aggregates and is no longer amenable to enzyme complementation when we add the tag binding protein. So we don't get fluorescence when there's insoluble aggregates. In the presence of a direct binding interaction with a ligand, that ligand will bind that protein target and stabilize it in a heat challenge, resulting in that 15 amino acid tag being exposed and available for binding with the microtag binding protein. So basically, with the thermal shift assay, we're detecting or quantifying the amount of protein based upon that interaction of that 15 amino acid tag with the microtag binding protein. A few more details about this system. To the left panel shows that our substrate is very versatile, and you can actually modify the output that you get from this assay system with respect to the wavelength. This is very useful if you want to use this system for imaging purposes or for some cell types that have autofluorescence or potentially some compounds that may be fluorescent. So there's versatility with respect to the output that you're going to get from this assay. You get to choose the donor quencher fluorophores, ultimately deciding on the kind of wavelengths that you want in the end. In the middle is a trace from a real-time instrument. So we've designed this assay system to work with real-time instruments such as qPCRs. These are very sensitive instruments, and what I want to show here is the dynamic range. For this particular target, you're going from, it's actually 12 million counts down to about 2 million counts. And what is going on here is you have a heating event. So you're heating the cells and assessing the amount of protein target there with each heat that you're applying. So it goes from 38 degrees at the very top, you get significant fluorescent signal, down to about 62 degrees at the very bottom, down to 2 million counts. So there's quite a large dynamic range due to the sensitivity of the instrumentation that this particular assay works with. To the right, just demonstrating that the reagents are actually thermally stable, and not surprising because this is an RNA, so they're very thermally stable proteins. And just to show that even above 60 degrees Celsius at the very end there, you only get less than 20% loss of activity of that microtag binding protein. So this makes it a very useful type of system for thermal shift assays where you're actually increasing temperature to cells. The workflow for microtag system is very simple, straightforward. It's a mix and detect reaction system. So the way it starts is to engineer a reporter cell line that expresses the microtag target. The microtag is that 15 amino acid tag. Grow the cells, transfer them to a PCR plate, add the inhibitor or activator or test ligand with the detection reagents. And then we use real-time instruments because we use the programmability of the heating and cooling steps of these instruments to test every well in a temperature series going from anywhere from 40 degrees up to 70 degrees. So the output to the right is an example of the type of result that you get from these real-time instruments where you're heating at a particular temperature and then detecting after each particular heat step to quantify the amount of protein remaining after that heat step. And this is more details about the heating and cooling programming of the microtag assay with these real-time instruments. So in the first step, you have the ramping to a temperature, let's say 40 degrees will be the first temperature that you want to test at, temperature 1. Hold for a few minutes and then drop the temperature down to room temperature to allow enzyme complementation and fluorescence to be generated. So now you quantify the amount of protein after that heat step at the second equilibration step. Follow this by the next temperature. Now you're raised to the next temperature. Now, let's say 45 degrees. Hold for a couple of minutes, drop back down to room temperature to allow fluorescence detection to take place, and so on. You can now go to a next temperature, temperature 3, maybe 50 degrees, drop it back down to room temperature, and look at the amount of target there after that particular temperature through the fluorescence that was generated. Now, it's very useful system with respect to you can program the heat steps for as long or as short as you like. You can extend or shorten the length of time for fluorescence generation after the heat steps. So it's a very useful system with respect to optimization for target. And anyone that's worked in cell target engagement knows that every target is different. And so this particular assay system is designed to allow flexibility with respect to the assay system, the time of heating, the heating temperature, the steps at which you want to heat, the amount of time after heating, how long does it take to generate fluorescent signal. And all this depends on the particular target itself. Some targets that are low or weak expressors typically take longer to generate fluorescence. Some membrane proteins you want to heat longer. So it's a very useful programming to optimize the assay for any particular target you have in mind. What I'm showing here is the raw data of what the output looks like on the left-hand side here. We're comparing a DMSO control with a test compound, the test compound in black. And on the x-axis, it says cycles, but this is really time. So each increment or each cycle is 1 minute. So after each heat step, there's a 10-minute detection of fluorescence. And what you can see here is that up to the 55 degrees Celsius temperature range, these curves overlap. But after 55 degrees, at the much higher temperatures, there's greater signal being generated in the presence of the test compound, which is indicative of a stabilization of this target with this test compound at these temperatures. Now, there's a couple of ways to analyze this data. One is the area between the two curves, between the black and red lines, would be an indication of how much or the degree of stabilization of this target by this test compound. The other is shown on the panel to the right, where we can look at the slopes of each one of these lines after the heat steps. It gives you an idea of the amount of protein still there after each individual heat steps. And this is very useful information, gives us an idea of the amount of the temperature at which the protein target is significantly impacted and how much it's impacted. So it gives us a quantifiable measure of the protein in the absence and presence of target at those heating steps. So just talk a little bit about the workflow and some of the data we generated for the Athos Therapeutics 91 program. When they came to us, they had an idea of some of the potential targets for this small molecule, but they didn't know what the main target or the main targets for this particular compound were. So we performed a target deconvolution in step one, where they provided us with a biotinylated version of the molecule. And it provided both the parental compound, non-biotinylated, and the biotinylated compound. So we used both of these in two tests, one where we had cell lysates treated with the biotinylated compound alone, and the second test where we pre-incubated with excess of the non-biotinylated parental compound, followed by the incubation with the biotinylated compound. Then we performed immunoprecipitation using streptavidin beads to pull down the biotin tag. So now we're pulling down that molecule and all proteins associated, and sent it for do wash steps and then sent for mass spec. And we performed a subtractive analysis. So as you can imagine, with the mass spec profile, well, we got thousands of proteins in the samples. But to sift through that data, what we did was to perform the subtractive analysis, where we were only interested in the proteins that were lost when we added 5 times excess of the non-biotinylated compound. And so we narrowed the search down to 153 protein targets. Doing further analysis of the data, doing a signal pathway assessment, and performing a more stringent cutoff of at least twofold change in the presence of non-biotinylated molecule, we were able to narrow down the target list to about 33 potential targets. So it's still a workable number, but still very high, using higher cutoff stringencies and incorporating that signaling cascade analysis. We then identified 6 strong potential targets of 91 from our mass spec data. So the next step was to generate the microtag reporter cell. So we generated 6 different individual reporter cell lines. And what was interesting about these 6 is 4 of the 6 were actually part of one signaling cascade and were actually part of a multi-protein complex. When we tested these 4 targets, so what you're looking at is thermal profiles in the black, and the top right is the target engagement with the parental compound. We're seeing target 1 and 2 both bind to 91. Target 3 doesn't bind. Target 4 binds. So there were at least 3 proteins here that were part of a signaling cascade that showed binding to 91. Now, it's highly unlikely that 91 was binding to all 3 individually, so we considered the possibility that they're forming a multi-complex together. They're in a multi-complex state, and then when we add the 91 compound to it, one of those targets is potentially being bound by the small molecule, resulting in a conformational change that may somehow indirectly impact the thermal melting or the of the other potential targets. So to separate these compounds, we want to do signal decoupling. And the way we went about this was we took the reporter cell lines and we performed immunoprecipitation with an anti-microtag antibody in high salt conditions. So with the high salt conditions, we wanted to strip away all protein-protein complexes that these individual targets were involved in, and then dialyze these to remove the salt, and then use the immunoprecipitated material for cell target engagement. What we identified was one out of these three targets at higher temperatures in the presence of 91 showed significant fluorescent signal or stabilization in the microtag reporter assay system, suggesting that this one target was actually being stabilized by 91 compared to these other two. And so we're looking at is 91 stabilization relative to 63, which is the non-specific compound. So it's it pointed us to one potential main target. It pointed us to the signaling cascade that this particular compound was impacting. So just to summarize, Solaris Bio, with our work with Athos Therapeutics, we performed target deconvolution and came up with a list of potential targets for 91. We used the microtag system as a way to validate the direct binding of 91 and really identify the main target of 91. And in the future, what we'd like to do, or what we have planned with Athos Therapeutics, is to perform an SAR strategy to test several advanced analogs that they have currently being synthesized, and test those with the different microtag reporter cell systems that we've developed during the course of the study. I just want to thank you for your attention and let you know we have a few more webinars coming up in the next year, so please stay tuned. We also have some workshops that we're trying to organize with respect to very specific case studies, and we'll try to make those much more interactive. And please feel free to reach out to us either through our website or to myself or Elmar directly, or Jordan. Thank you very much, and I think we have time for questions.
I believe so. So we are on time here. So let's answer some questions here. Please go ahead and drop your questions in the chat. And I do see some questions here. The first one is for Jordan. If you have presented a drug discovery process starting from AI and all the way to preclinical development. How can your experience now be translated and benefit other AI-based drug discovery projects in other companies?
Oh, that's actually a multi-sectional question, because by participating, I would say, drawing from my experience in participating into driving the creation of the platform, I had to really think deep, because I was also being used as a beta tester, basically, for that platform. I had to look deep into what was important in finding good targets, and what were the details, the little properties of those targets that could make a difference. Like identifying networks is one thing, but really choosing the targets that you're going to go after, it still comes down to personal expertise and knowledge of the biologist that is handling these programs. So I would say it really, really drove me to look into myself and really bring to the forefront what I think is driving my decision making. So basically, it made my decision making materialize in front of me. So I think that what it did for me is I can look at information right now and process it faster, since I had to look really deep into that decision making. Also, I learned a lot on how much to trust the systems and how much to use my own input and my own intuition. So these systems can offer you certain uh, suggestions. However, you have to do your own thinking and your own deep search after that. But what they do is basically categorize this information, put them in order, and we try to create a platform that all the questions that we would have as biologists,
Mm.
it would make them available, it would make the answers available by a click of the button. So this created also this platform in my head where I immediately ask the relevant questions about the compounds or the pathways that are important in going to the clinic. So I
Jordan,
guess it
we have
was a practice. Yeah, it was like, practicing on how you use your own words. Like my biological vocabulary that was in my head materialized, and now I can bring it forward faster.
Jordan, thank you. We have a follow-up question for you. So, talking about the platforms, drug discovery platforms, what is the ideal integration of the microtag technology or the workflow into drug discovery processes and programs, in your opinion?
Uh, I don't know how deep Ivan went into the high throughput capabilities of these models. Here, we are not talking about anything. Actually, the technology by Solaris is pretty new, but the ability to use it in a high throughput manner is what makes the difference. What our platforms do is they integrate all this information and they shrink time. This is what the platforms do. Instead of having to spend all your time to come up with these pathways, with this background information, with a click of a button, everything can come to your screen. But certain steps, like identifying the target and looking at many different, identifying target is one thing, but as everybody in that drug discovery knows, there are also platforms of chemicals. There are some chemicals, when you're doing your SAR, there are some chemicals that are better than others. So there are many factors that play a role here. And high throughput capabilities of analyzing these characteristics of these compounds are very important in shrinking the time. Actually, what we did with these types of approaches is we went to the clinic from target identification all the way to the clinic in 2 years by using such systems. So, I mean, you need certain type of luck, but these types of high throughput approaches reduce the requirement for luck.
Right.
I mean, it basically enhances your ability to use more information to make a way more well-informed decisions.
Now, we certainly want to reduce the luck factor during drug discovery. That's for sure.
Oh yeah, and everybody knows, like, we want to take credit for everything. These types of approaches reduce the luck factor. And like I said, the high throughput capabilities that can be combined here is basically combining one high throughput type of analysis that was slower in previous years to a high throughput type of analysis that became faster earlier.
Right. Jordan, there are more questions for you coming in. So, since we have a little time left, so let's go over those questions. So, one anonymous attendee is asking, was the target present in the previously tested panels?
Excuse me?
Was the target present in the previously tested panels?
No, it wasn't. In the previous tested part, it wasn't present because we were not looking at all in these types of targets. Listen, we are biased now. This is how important the high throughput part of this is too. We are biased in our approaches. Whether we want it or not, there is certain type of bias based on the type of success we had in the past or the type of research that we did in the past.
Right.
When actually this target came as a great, actually differentially expressed gene
Huh.
in many of our analysis for our lead compound. But we never, it was a great target, actually, that we never would have looked at. We were not
Good.
looking in that direction at all. Actually, it
Here's
blindsided us.
another question for you, which is, a follow-up type of question. How did you move your compounds into development of IND candidate based on your AI and phenotypic cell screening scores? Question mark.
Ah, that helped. Listen, our AI helped in identifying the target. Now we are actually developing it to do more. It's still, the platform is still in development. It's not out there.
Hmm.
We are still adding abilities to it. We are adding clicks there where you can bring either types of projections, let's say like chemicals. We're trying to bring programs that can fit chemicals into certain enzymes that are identified by our platform that can do the jobs of many, many analyses in one platform. However, we identified the target, and everything after that went through more high throughput platform like yours, not for the primary target, but we went high throughput in some analysis of safety. We tried to do some high throughput analysis in our characteristics of the compound, the chemical characteristics of the compound. But we always used these platforms that are out there also to predict actually chemicals that fit in a certain target as a guide. Everything was, in the end, everything was proven in the lab. And the ability to do things in a high throughput manner in the lab is, I mean, it's immeasurably beneficial, I would say.
Thank you, Jordan. So, last question for you, Jordan. A user, an attendee is asking, then you worked backwards to identify the target?
Yes, not by choice. Like I said, and I'm giving you the story behind this is like we outsourced this particular assay. It wasn't very high throughput, so it was taking time. So we outsourced many, many molecules for backups. But 91 was by far the best. Well, not many molecules, but some. But the lack of high throughput in that particular manner delayed. We were confident it's hitting the target, and we were surprised by it. And now we
Great.
have another program. This is
Jordan,
where
thank
luck
you. I have
plays a
a
role.
few questions lined up for Ivan. So, one question is, how repeatable, reproducible is microtag success with respect to other targets? What target families mostly benefit from microtag?
Yeah, we want to focus this assay for challenging targets, right? For well-behaving targets, there's other ways that you could do cell target engagement, other ways that you could assess drug binding, for example, SPR. So what we're focusing this assay around is these challenging targets like membrane proteins and transcription factors, which eventually will be having workshops around. And so these are the difficult targets that you can't express outside of the cell. They require a cell environment. And these are the ones that we're developing numerous case studies for. And we're designing the entire assay system around to try to target these challenging proteins. And then with respect to reproducibility, every target is different, right? I mentioned it earlier. Membrane proteins do not behave the same way as transcription factors, and even individual membrane proteins, right? They're going to be very different. Is it a multi-pass, single-pass membrane proteins? So all the assay system has to incorporate various optimizations whereby you could be confident and have significance with respect to seeing a stabilization or a destabilization of the particular protein. So, answering the question, you have to set up the optimizations to make it reproducible. We have a number of in-house uh, tricks, and we have some inhibitors, some compounds that we use as reference compounds to work with some of these challenging proteins. So yeah, we can make it work. We'll figure it out. There's various ways to optimize for each one of these types of proteins.
Ivan, I know we're at the top of the allocated time. If you can quickly answer these two questions, that would be great. Ivan, has Celaris Bio used the microtag technology to interrogate compounds that cause a destabilizing instead of stabilizing aggregation of temperature?
Absolutely. So the majority of the compounds will stabilize, but we do come across destabilizing compounds. And with the destabilizing compounds, rather than seeing an increase of fluorescent signal above the DMSO control, you start to see a decrease of fluorescent signal. So we see that about 10% of the time when we work with compounds. And what's interesting is we have proteins that we've worked with that it depending on the site where a molecule binds, it will determine whether it is a stabilizing event or a destabilizing event, right? We can have one molecule binding protein A, causing it to destabilize, and another molecule binding the exact same protein but on a different site, it now stabilizes. So we've got, we can identify both stabilizers and destabilizers with the assay.
Great. Along those lines, the same attendee is asking, can Solaris Bio expand the advantages of can be it advantageous of using the microtag for multiplex measurements at multiple temperatures versus sets a hybrid where you measure only at single temperature? Can you please
Absolutely.
briefly comment on that?
Yeah, that's why we have our assay system designed with real-time instruments, so qPCR real-time instruments. Now we've incorporated that heating and cooling capability of these instruments to span through all different temperatures that could be impacting the thermal stability of that protein. So sets up you need to go through a thermal profile. You identify the temperature of melting for that particular target, and then you can interrogate that target at that 1 TM temperature. What we're doing with this assay, eliminate the need for identifying that temperature of melting and incorporate various heating steps that are going to impact that temperature. And that's why I'm saying our typical scan or a typical read is going to go from 40 degrees up to about 70 degrees. And after doing so many proteins, we know that the majority are going to melt between 45 and 65 degrees. The majority of proteins. So we don't miss anything. And so you're also not biasing to one particular temperature. What we're noticing when we do a scan and then do an analysis of the amount of protein target after each temperature is that it varies. There's potential binding events that are happening at temperatures above the melting temperature, and some binding events that are happening at temperatures just a few degrees below that melting temperature. So we're not biasing to one particular temperature when we're assessing the binding of a drug to a protein target.
Well, thank you. Please get back to us with more questions. I want to just make a comment about the future workshops. We will come back with a series of workshops and we will separate them by target families. We will separate them by modalities and various workflows. We will also open up the workshops and we will take input from potential users as well. So we will reflect onto the needs and basically we will come back with workshops that will really give you something that is going to be useful in your drug discovery programs. So stay tuned for future workshops and webinars. And thanks again for joining us today. See you next time. Thank you, Jordan. Thank you, Ivan.
Thank
Thank
you.
you, everybody, joining us today. Bye.